Welcome back to CNN, Cancer News Network. Evelyn Howell here. So let's get back to exactly what is causing colon cancer at the molecular level. Dr. Lowe?
Well, Evelyn, to put it simply, colon cancer arises from tumors in the colon. Tumors are masses of cells that are abnormal in size, appearance, and quantity. What causes this abnormal growth? Mutations in genes. Let's take a closer look. All cells in your body have 2 copies of each gene. Genes are made up of DNA, which is made up of base pairs. These base pairs you can think of as letters representing molecules, and each letter also has a different letter that it likes to pair with. The DNA is read by something called a polymerase that is brought to the DNA by a transcription factor. Once the transcription factor has attached the polymerase to the polymerase can begin to move along the DNA. You can think of this polymerase as a machine that traverses the DNA, producing a new chain of corresponding letters. This new chain is known as RNA. This RNA will then be turned into a protein. A protein is made up of amino acids, and each amino acid is coded for by 3 letters of RNA. So DNA, which is made up of base pairs, can be turned into RNA, which is made up of corresponding bases in a process called transcription, and RNA can be turned into a protein, which is made up of amino acids in a process known as translation. You probably already know that there are a bunch of proteins in your body. Each protein has a specific function or task. Some proteins regulate cell division, some are involved in the 2 processes that we just talked about, transcription and translation, while some proteins even activate other proteins.
So what does this have to do with colon cancer?
Well, let's think all the way back to DNA. What if something was wrong with your DNA?
Well, that would mean that the RNA made from your DNA would be abnormal, and then that the protein made from the RNA would also be abnormal.
Exactly. And having abnormal proteins means that they cannot accomplish their tasks, causing major problems, one being colon cancer.
So how does something go wrong in your DNA?
Something I'm sure that you've heard of before, mutations. In colon cancer, the most common mutations are nonsense and deletion mutations. A nonsense mutation is a change in a base pair that leads to an amino acid that stops translation. A deletion mutation is when one or more base pairs are removed from the DNA, leading to a change in many amino acids, one of which also stops translation. Both of these result in a shorter protein than normal, which prevents the protein from functioning properly.
So I'm still a bit confused as to what all this has to do with colon cancer.
Well, let's go into the body and see for ourselves. This is what the cell looks like that we're going into, just so you know. The large green circle represents the cell membrane, the red box represents the proteasome, the red tower represents the nucleus, the blue circles represent beta-catenin proteins, and the red circles represent APC proteins.
Whoa! What's happening? You're becoming beta-catenin. We're headed straight for that intestinal cell. Quick, grab hold of this green stuff. It's the outer shell of the cell called the cell membrane. Don't let go!
Wow, what just happened?
Well, you just became beta-catenin, a protein located in the cell membrane that allows cells to stick to each other. Now, you see those red guys floating in the liquid stuff called the cytoplasm? Those are APC proteins. Don't go near them. They'll bring you to the destroyer, Lord Proteasome.
Well then, why do some of the beta-catenins ever let go? Like that guy.
Because some try to make it to the nucleus to find their soulmate, a transcription factor.
Is this what happens in all cells?
Well, only if it's functioning correctly. Let's check out the cell next door.
I thought you said we couldn't leave the cell membrane. Why are there so many beta-catenins floating around in the cytoplasm?
Well, you see those red APC proteins? Look how dysfunctional they are. There's no way they can reach out and grab you to bring you to Lord Proteasome.
So we can just let go?
Let it go, can't hold it back anymore. Let it go.
Whew. Wow. So what was wrong with the APC in the tumor cell?
Well, remember that before APC was a protein, it was first a DNA, and you recall that DNA can have mutations causing problems in the protein that's made. Well, the APC DNA that made up the APC protein had mutations. Most likely these mutations were large deletions or nonsense mutation, which remember leads to a dysfunctional protein. Because the APC was dysfunctional, the beta-catenin from the cell membrane was able to enter the nucleus as shown in a published clinical study by Rensky-Sheenstra, a doctor. Once beta-catenin is in the nucleus, it can bind to a transcription factor, leading to transcription of genes that will ultimately regulate cell division. This was shown in a different study by Hans Clevers and his colleagues at the Hubert Institute. Together, these studies show that in colon tumors, the inactivation of APC allows beta-catenin to enter the nucleus, bind to transcription factors, cause transcription of genes that will become proteins that regulate cell division, and together these cause tumors.
Wow, that's fascinating. Very insightful. This concludes our segment on colon cancer. Join us next time for Show and Tell with my pet African parrot. And Dr. Lowe will join us once again to finally reveal the relationship between population genetics and the mysterious bean. This is Evelyn Howell, signing off.